Hi, it is me, Dr. Ghulam Yaseen from the University of Education, Lahore, Pakistan. Let's start my introduction. I am working on the medicinal plants and their different types of the biological activities, including antioxidant, antibacterial, and other pharmacological activities. Basically, recently we started a new project that was confined with testing the medicinal plants against antibacterial activities. Recently, we modified it and we synthesized some sort of the nanoparticle from the selected medicinal plants, and we tested their antibacterial activities and further their testing using some sort of in silico studies. Basically, the today presentation is confined. Its title is Medicinal Plants Mediated Engineered Silver Nanoparticles and Evidence from the In Silico Approach. Basically, in this study, we selected medicinal plants and we engineered the silver nanoparticles and we tested their antibacterial activities, and further the antibacterial activities or antibacterial potential was confirmed using in silico approach. Let's start our presentation. This is the table of content of today's presentation, including its introduction, objective, methodology, results, conclusion, achievements, and references. Basically, when we are going to discuss about the nanoparticle, we should have information about the nanoinformatics. What is the nanoinformatics? Basically, the nanoinformatics is a multidisciplinary approach that focuses on the development and application of the informatics tools and different techniques and different approaches in nanotechnology. And there are the different fields of the nano research, nanoinformatics, and different its importance and different its applications. Why nanoinformatics in drug designs? Basically, it is the precision medicine targeted delivery when we synthesize the nanoparticles using nanoinformatics approach. And when synthesized nanoparticles are achieved, they have the enhanced bioavailability. And we study the systematic toxicity using nanoinformatics approach. And using the nanoinformatics, basically, we can overcome certain types of the problems that are faced in nanotechnological studies. What are the silver nanoparticles? Why we selected the silver nanoparticles? One of the importance is they have the more enhanced performance and they have the precise targeted delivery. And they have reported effective antibacterial potential, and many protective interaction controls can be studied using silver nanoparticles. What are the objectives of this present study? Our first objective was to synthesize the nanoparticles using a selected medicinal plants, that was the Calendra heracei leaf extract. And second objective was to characterize the nanoparticle that was synthesized using selected medicinal plants. And other was to design the structures of the nanoparticle that we have synthesized. Another was that to interpret the effects of silver nanoparticles on the genes of the Staphylococcus species. Basically, we have discussed that we tested that silver nanoparticles and we have confirmed their action using in silico approach. And during in silico approach, we perform in silico studies using the selected genes of the bacterial strains. This is the overview of the methodology. how we worked on the medicinal plants. First of all, we selected the medicinal plants, we prepared their extract, we performed their phytochemical studies, we synthesized the nanoparticles, and then we characterized the nanoparticles. And then after characterizations, we perform nanoinformatics studies, including the design of silver nanoparticles and their production using zeta potential nano extract. There is a software. We perform the cytotoxicity studies using a software using cytox techniques, and we retrieved the selected genes of the Staphylococcus species and their further docking studies and molecular simulation. And this is the fourth section of our study, that is the results and analysis of the present studies. It includes the synthesis and their characterizations and their docking analysis and their toxicity analysis. First of all, we prepared the plants, we collected the plants for studies, collected from the botanical garden, and its washing, its grinding, stirring, filtration, and storage. And after this, the plants, we perform the synthesis of the nanoparticle using the leaf extract of the selected plants. And this is the overall pictorial view that that elaborate or that express the synthesis of the silver nanoparticles. And this slides especially is a pictorial overview of how we characterize the nanoparticles that we synthesize using silicon. First of all, we perform SPR analysis and then perform the scanning electron microscopy to study the morphology of the synthesized nanoparticles. And beside, we perform the FTIR analysis to confirm the different types of the functional groups present in the synthesized nanoparticles. And further, we perform EDX analysis to confirm the surface components of the nanoparticles, and to confirm the nature or to elaborate or to express or simply we can say to analyze the nature of the synthesized nanoparticle, we perform XRD analysis. This is the slides that show or that confirm the synthesis of nanoparticles using SPR analysis, or simply we can say we perform UV spectroscopy, spectrophotometry, we can say. And this show our nanoparticle, synthesized nanoparticles show the range between 200 to 500 something wavelength in nanometers. And this is the slide that confirm the morphological size or morphological shape of the characterized nanoparticle, synthesized nanoparticle using scanning electron microscopy. This is another parameter that predict or that show the different types of the functional groups present in synthesized nanoparticles that we confirm or that we perform using FTR analysis. And during FTR analysis, we observe that what type of the functional group are present in synthesized nanoparticle. Basically, that functional groups protect the certain types of the compounds present on the surface of the nanoparticles. And this is again show the XRD analysis that predict the nature of the characterized or synthesized nanoparticles. And now come to word the applications of synthesized nanoparticles against selected bacterial strains. Our goal was to determine the antimicrobial activities of the selected samples. We performed the dose 500 microliters 2500 millimole and we selected the 500 microliter of the nanoparticles, and they performed the molar solutions and 500 microliter of the plant extract. And we performed the antibacterial activities using the disk diffusion methods. And during disk diffusion methods, we observed the zone of inhibitions. And these slides show the zone of inhibition that show that how much nanoparticles inhibit the growth of the selected bacterial strains. After performing the antimicrobial activities, we confirmed the antibacterial potential of the plants using the nanoinformatics approach. In nanoinformatics approach, first of all, we confirm the 3D structures of the synthesized nanoparticles using Avogadro tools, and then it was observed its potential for the using the zeta potential, and using zeta potential, we observed the value of minus 23.55. That is the more reliable value in the zeta potential. And then we checked the cytotoxicity analysis using cytox approach, and its value was found to be 90, and that 90% cells were viable. And then we selected the genes of the Staphylococcus species using the AlphaFold techniques. And these slides show the selected genes of the bacterial strains where we want to test, where we want to confirm that our nanoparticles affected which type of the genes. And these slides show the docking analysis, where we selected the ligands of silver nanoparticles, and ligand of the silver nanoparticles affect work on the genes of the selected bacterial strains. We use the Swiss doc tool and in Discovery Studio. And this is the overall ligands and interactions between the ligands and the selected genes of the bacterial strains. And this is the QSR analysis that show that silver nanoparticles contain some sort of the compounds that affected, or that shows some sort of the effects on the genes of the selected bacterial strains. This is the different types of the scores that we can be discussed. And these slides show the what type of the toxicity analysis. It shows the synthesized nanoparticles are not toxic. It can be used in the drug discovery developments. And this is the further dynamic simulations using different types of the approach, RMS deviation plots, RMS fluctuation plots, and plot of the hydroids, simply we can say, plot of the various introductions, radius of the gyrations, and solvent exiguity, we can say. That is shows the dynamic simulations in our studies. That was the focused. Next, come to the conclusion of our study. What we have concluded. We have concluded that the key characteristics of the synthesized nanoparticles have shown the successful strategy for the future drug-resistant Staphylococcus species analyzed through nanoinformatics approach. Overall, this study show that how a selected medicinal plant can be used against antibacterial potential can be studied in antibacterial studies, and further their antibacterial potential can be enhanced by synthesizing the nanoparticles. And when we synthesize a nanoparticle, we should first of all, we should characterize them. And that characterization shows a successful synthesis of the nanoparticles. When characterization is done, then nanoparticles are tested against your targeted studies, such as antibacterial studies, such as antifungal activities, or anti other microbial studies. And another question arises, what type of synthesized nanoparticles are either toxic or not toxic? We should perform some cytotoxic studies. When you perform cytotoxic studies, it shows either your synthesized nanoparticles are toxic or not toxic. And further, when you have confirmed your medicinal potential of the selected nanoparticles or synthesized nanoparticle, it is new or recent approach to predict their targets, how they targeted the bacterial strain. In the present studies, we targeted some sort of the genes where we predicted that our synthesized nanoparticles have potential to affect certain types of the genes, and where that genes will are the responsible for the making the cell wall or other metabolites in the bacterial strains. When that genes are ineffective, definitely that bacterial strains will be killed. Thank you so much for your patience, and thank you so much for the organizers who organize these events and organize this conference. And I hope that this conference is found to be more effective for the different uh, platforms or different scientists from the different regions of the world. And let's coordinate with each other. Let's collaborate with each other for the future studies. If you want to collaborate with us, you can contact with your conference organizers. And thank you so much.